So when Garrett was a postdoc with Young Dan at Berkeley, Young's lab and my lab had very similar perspectives at that time. And Young had also done a lot of natural images stuff, kind of coming at it from an independent perspective, but we were both doing a lot of natural images. And actually, I think Garrett's very first neuroscience paper was the decoding paper, right? So if you want to see one of the first awesome vision decoding papers that was ever published, it was Garrett's 1999 paper in Journal of Neuroscience. It's a really cool paper, and he probably, since he's doing like somatosensory stuff now, he won't actually say anything about that paper if he teaches a course. I don't know, do you ever mention that paper? You should look at the paper. It's a really good paper. And basically all of the visual decoding stuff that anybody does kind of builds on that paper. It's a foundational paper. Thanks very much for having me. I think it's wonderful coming to engineering neuroscience groups. I think neuroscience desperately needs engineers, and I'm really happy that Georgia Tech is building up in this area. And the reason is, before that, let me get to the meta issue, which is you may have noticed if you happen to read the abstract that it seemed kind of schizophrenic. And that's because I thought there's a bunch of neurophysiology people here and there's a bunch of MRI people here, and I was trying to figure out how to make both groups happy. So I decided I'm just going to give 2 talks. The first half of the talk is going to be about neurophysiology. The second half of the talk will be about MRI. But hopefully you will see that although these are 2 very different approaches on 2 different primate species, there's a common theme, and I'll begin the talk with that common theme, which is really about reverse engineering the brain. So I'm just going to bore you a bit with my personal philosophy. And it builds on kind of this observation, which is you can think about neuroscience rather than as science as a reverse engineering problem. In engineering, usually we have a model, some mathematical theoretical model with well-characterized limits, and we try to build a machine that matches that model as closely as possible. And in reverse engineering, we have a machine that somebody gave us, we have no control over it, and we try to build a model that describes that machine as closely as possible. And that's reverse engineering. And these both have the same kind of components. You have some sort of device. You have a measurement apparatus. It's going to measure the device, make sure it's in spec or figure out how it works. And you've got some sort of modeling framework. But the ways that these 3 factors get mixed together in these 2 approaches to engineering are different. And the constraints in these 2 directions are very, very different. And in particular, in reverse engineering, our limitation is usually in the amount of data we can gather to characterize the system. And in neuroscience, that's definitely true. The real problem we have faced today in neuroscience is measurement. It's all measurement limited. Now, if we could record from every neuron in the brain for days and days and days, we would have a different problem, which we would not know how to model that data. But that would be a really good problem to have. And we're not even at that problem yet because we can't even get the data we need to model this complicated nonlinear dynamical system that we have in the brain. So I tend to think of all of this, since it's a reverse engineering problem, I tend to think of it as an optimization problem. I've got a complicated nonlinear dynamical system that I'm trying to model. I want my model to predict how the system will respond under new conditions. I want it to generalize. I want my model to predict as accurately as possible what data do I have to collect given the constraints of the methods I'm allowed to use, and how much data do I have to collect in order to build the best possible model. And that's not like the perspective you go into if you're designing experiments with this in mind is not kind of a point null hypothesis testing perspective. It's more of an optimization perspective. So everything in my lab I tend to think of as an optimization problem. And in particular, I tend to think of this as kind of trying to maximize the mutual information between the source, which is my brain, and the destination, which is my computer. I want to get as much entropy from this brain over here into this computer so that I can basically build a model of the brain on my computer. But the problem is, to go from this source through this destination, I have to go through some channel. And this channel, just like any information theory system, this channel is lossy and limited and noisy. And in fact, all of the measurement methods we have for neuroscience are basically bad. They're all really bad. They're just bad in different ways. So you guys have probably all seen this graph before. This is a description of the different methods we have available for neuroscience, where we have the spatial scale of the measurements and the temporal scale of the measurements on this graph. And all of these measurement methods at the bottom are methods we're allowed to use in animals. And a lot of these are really good. They have really good spatial resolution. They have really good temporal resolution. And of course, the wonderful thing about the rodent models over the last 10 years and the evolution of measurement is that we keep developing new methods of measuring more and more neurons from rodents over time. And then up here at the top, we have all of the methods that are available for humans. And all of these methods have problems. Some of them are fairly fast, but they have really low spatial resolution. Some of them are fairly slow, but they have excellent spatial resolution for humans. But excellent spatial resolution for humans means 2 millimeters, not a membrane. So all of the measurement methods we have for humans are going to be bad. And we want to pick the measurement method that essentially will optimize our information flow through our experiment. Then the other thing we have to think about if we're solving this optimization problem is what kind of experiment are we going to do and what kind of modeling framework are we going to use. And as Garrett mentioned, I've always thought about all of this reverse engineering problem as a black box problem. This is if you remember, for all those of you who are engineers, you took like a 3-course sequence, some variation of a 3-course sequence. The first course was linear systems, and they told you, here's a black box, but it's linear. How do you characterize it? You get the impulse response. It does everything. Yay, great. And then they go, OK, next course, nonlinear systems. How do we deal with that? Well, you can kind of like project it into this space where even though it's nonlinear, as long as it's stationary, you can still treat it with linear systems. Linear systems, yay. And then you take the nonlinear dynamical systems course, and they go, ooh. Well, we have no idea how to do this. Let's pretend it's linear, yay! And everything works. So linear system identification is always about somehow like linearizing your system so that you have some hope of getting something out of it. And that's a perfectly valid thing to do. So this is kind of a way to think about this black box problem, this system identification problem, in a statistical point of view. I've got some x variables, which are either my stimulus or my stimulus at a task is also an x variable, right? And then I've got some y variables, which are the data I measured. And I've got my black box, and I'm just going to measure it using some regression model, right? This is a regression problem. It's a giant regression problem. I can either treat it from an inference perspective or an optimization perspective. In practice, optimization ends up being the best thing to do because linear regression or linearized regression is a really great thing really great method that works really, really well. And so we collect some data with a fit set, and then we collect some data with a test set, and we just look to see how well our model predicts in the test set, hopefully using new stimuli and new tasks that we didn't use to fit the model. And eventually, we're going to hit the noise ceiling, which is the best possible model we could ever build, given the data we have, and then we're done. Then we need to go get some more data. So think about all this optimization perspective. This means that you're going to design the experiment using a very, very different method than is normally used in biology and psychology. Normally, all biology and psychology is built on point null hypothesis testing. Point null hypothesis testing you can think of as binary gradient descent, but it assumes that the local hillside you're on in this state space of possible models is the right hill. And my complaint is, given that if we had every neuron in the brain, we wouldn't know how to build the model of the brain, it's unlikely that any hypothesis we have about the brain is even close to the optimal hypothesis. And the problem is if you end up pushing a point null hypothesis paradigm really far, and eventually it breaks, and your null hypothesis cannot be rejected, you have no idea what to do, because you've only mapped this local part of the gradient of the entire model space, the entire hypothesis space. So I prefer kind of a data-driven method where we sample as much as possible broadly across this space in order to sample as many hypotheses as possible and as many possible model forms as we can as rapidly as possible. And if we're doing that, then we're going to select the method that maximizes the mutual information through this system per unit time, money, graduate student. That's really the factors that limit science, right? Time, money, and graduate students. So per unit time, money, and graduate student, we just want to get as much data through entropy through the system as possible. We want to optimize our experiment to maximize the information over the hypothesis space. So not just one hypothesis, but all possible hypotheses that might be relevant. We want to gather data that spans all of them. We want to fit an explicit quantitative computational model that actually makes quantitative predictions on the dependent variable, whatever we measured. We want to make sure that this is cross-validated so that we have a fit set and a test set that are separate, that are two separate things, so that we make sure that we're not overfitting. And we want to always measure our cross-validated prediction relative to the noise ceiling. The noise ceiling is given by the intrinsic noisiness of our measurement and how many data samples we have. And we want to test generalization of the model under naturalistic conditions. So there was a giant war in the vision community that was fought in the 1990s about whether we should understand vision like television engineers and just use sine wave gratings for everything. That was like the classic view. And then you had this crazy community of people that said, no, this is a nonlinear dynamical system. You have to measure it under the conditions that it operates. Because you're going to build a model that's not perfect. You want at least the nonlinearities in that model to be the valid nonlinearities that the system operates with every day. And after 10 years of fighting, the community kind of concluded, OK, you can build your model however you want. Doesn't matter. You can build it with gratings. You can build it with natural images. You can build it with dots. Doesn't matter. But when you test it, you have to test it under naturalistic conditions. Because the only way to know if you have a nonlinear dynamical system that your model predicts well is to measure that system and predict in the situation where that system normally operates. So I would encourage you guys to do that if you're not doing it already. Certainly number 5, no matter what you do, I think number 5 is really, really important. I should mention in biology and psychology, almost nobody ever tests predictions. And almost, I would say like 95% of people don't test predictions, and 99% of people don't test generalization. And that's because in this field, everybody does point null hypothesis testing. Everybody focuses on statistical significance. Okay? This is my little rant about how much I despise statistical significance. I taught statistics for 15 years at Berkeley. I kept trying to discourage students from even thinking about statistical significance. Statistical significance is necessary but not sufficient for doing science. Statistical significance is just a really bad 1930s method for trying to get some sense of whether your data are random or not. And trying to move science forward by finding non-random versus random data sets is not a very good way to make progress, right? So I would encourage you build predictive models. All right. OK, so now I'm going to talk about physiology. How long did that rant go on? 15 minutes? Did I talk at 11:15 or 11:20? I get till 12:15, right? OK, I'll talk fast. So let's talk about neurophysiology. The rest of my talk will take 5 minutes. No, let's talk about neurophysiology. This is a macaque brain. We flattened it out here just so you can see all the little brain areas. And of course, as you all know, the macaque is a hierarchical visual system consisting of something probably on the order of 40 visual areas connected in this very rich, complicated network. And for many years, probably the first 20 or 30 years of my career, I focused on this little blue area here called area V4, which is an intermediate visual area. It's not primary visual cortex V1. It's not inferotemporal cortex, where you already have neurons selective for objects. It's somewhere in the middle. And the reason I was focusing on that area was I thought, well, it's not as boring as primary visual cortex, but it's not hopelessly complicated. Maybe by the time I'm dead, I will be able to figure out what this visual area actually does. And the first part of this talk is, I think maybe I've kind of figured it out, and I'm not dead yet. So that's a success. All right. So what does V4 code for? Well, here are some classic experiments on V4. This is old data that I published showing that this is a one V4 neuron on the left, one V4 neuron on the right, and this is just the firing rate to these different little stimuli called non-Cartesian stimuli. And you can see that this neuron really cares about these hyperbolic gratings. Maybe it's interested in intersections. Maybe it represents intersections. This neuron cares about curvature. Ed Connor, my colleague from David Van Essen's lab, went on later to show that these neurons can be selective for little corner elements of shapes, in particular these little extruded points and pieces of curvature, little local sort of contour elements of shapes seem to be represented in this area. But after 30 years, I would say, of work in V4, we really have no V4 model. Oh, wait, sorry, one more thing I should mention. There's not only neurophysiology data to indicate that V4 represents these intermediate shape features, whatever the heck they are. There's also some indirect evidence from modeling using deep neural networks. So on the top here is a standard deep neural network you probably have all seen. This is like the original Café network, I think. That's just a hierarchical network trained on natural images. So it somehow embodies the statistical regularities of natural images, because otherwise it wouldn't be able to do image classification. And on the bottom is a human data from an MRI experiment We're looking at the back of the head where the visual system is in humans. And now we've colored each of the little locations in the human occipital cortex according to the level of this cafe network that best describes that location. And you can see that these early areas in primary visual cortex are all described by very early layers of the network, COM1 and COM2. And the highest levels of visual processing in the human, which are these areas you may have heard about if you ever went to a psychology talk, the occipital place area, the extrastriate body area, the fusiform face area, things like that. Those are all best modeled by these really highest levels of the network. And the middle areas like V4 are represented by these intermediate levels. So there's a lot of indirect evidence that this V4 area, just as you'd think based on the visual hierarchy, is representing features of intermediate complexity. But the question is, what are those? And the bottom line is we do not have a good model for V4. This is the best model for V4 that's out there. This was published back in 2006. And this basically says a V4 neuron takes the image and it calculates the power spectrum of that image. And it has a tuning curve based on the power spectrum. That is an egregiously stupid and brain-dead model. There is no possible way that this model could be right because obviously the visual system has to encode phase information. Otherwise, everything with an identical power spectrum would look the same, and you wouldn't be able to tell the difference between Bilal and a cloud, which would seem kind of bad. So this model has to be wrong. Well, can we do better than this? So what we decided after many years of hitting our head against this is we just did not have enough data. We realized these are awake macaque physiology experiments, and we realized that we could not possibly get enough data in a day. We would need, from a neuron, in order to characterize the receptive field, we would need, say, 200,000 to 500,000 frames of video to be able to even make a stab at characterizing a receptive field. And that would be hours of data, and we would need to record over multiple days and weeks. So we started implanting these Utah arrays in area V4. And these Utah arrays are awesome because they stay in there for weeks and weeks and weeks, or months, or years if you're lucky. And you can record continuously from the same neurons over many days. Now, if you look at the action potential of the neurons across days from one of these electrodes, they will actually change. And that's because the physical location of the brain is moving slightly relative to the electrode. But my lab, and independently Jim DiCarlo's lab at the same time, came up with this cool method that we call the fingerprint method. And the idea is you bring your animal in every day and you show him a movie. In this case, this is a movie that's 450 frames long. And you just map the peristimulus time histogram across during this movie across days. And even though the waveform of the action potential projected onto the electrode is slightly changing from day to day, the neuron's selectivity for these complicated natural movies remains invariant. And then you can actually map across days one electrode, and every single neuron has a unique functional fingerprint that reflects its stimulus selectivity, and it doesn't look like any other neuron in the brain. And so you can track these neurons over days. You can see here we have one neuron for 37 days, and it's exactly the same neuron. That meant that we could collect millions of frames of video from each neuron. So a typical neuron in this data set, we have like 800,000 frames of video. Some neurons we have 3 to 4 million frames of video. And my general rule of thumb for life is if I can't figure out what a neuron does after 1 million frames of video, I'm going to give up and go get another job. So we have 800 of these neurons. We have a lot of data from each neuron. We should be able to finally solve this problem. Now we have a big data problem. Oh, I should mention the stimuli we use. We use simple stimuli and we use natural movies, just so we had both. OK, so now we have to build a model. We recorded in 3 areas, V1 and V2, where we accidentally ended up exactly in the fovea. So as far as I know, these are the only primate foveal recordings made in an awake animal. And the reason we could do that is even though there are microsaccades that are fairly large, because we have infinite data, we can basically aggregate over those microsaccades, and we can model them out. We also recorded from V4. So let's start with V1 and V2. As you probably all know in here, V1 and V2 can be neurons. V1 neurons can be described as these spatiotemporal Gabor functions, and that's because any natural image can be decomposed into a linear superposition of spatiotemporal Gabors. That's just a feature of natural image statistics. So we decided we would basically start with this sort of Gabor model. And we have a simple cell model, which is just a Gabor, and then halfway rectification, or a complex cell model, which is 2 Gabors, squared and summed, and then rectified. These are completely standard models. And these are going to be the elements of our sort of network model. But we're going to incorporate other things too. We want to deal with color, so we have to deal with LM cone opponency. We think that these receptive fields are taking spatiotemporal derivatives, so we want to incorporate that. We want to deal with all of these things somehow or other in our model. So rather than building this model explicitly in terms of kind of a classic engineering approach of putting little elements together, which we've done, we decided in this particular experiment to take advantage of the fact that we have GPUs now and things like PyTorch and Keras that allow us to do gradient descent using deep neural networks really, really quickly. But rather than just building a generic deep neural network, this is an architected deep neural network. It's very, very specifically designed to both incorporate all the assumptions we know from previous V1 physiology into the model, and it's designed to be able to learn on a fairly small data set, because even though we have 1 million frames of video, it's small relative to a data set you would normally train a deep neural network on. So let me just do it graphically here. We've got our stimulus. The stimulus gets moved into a bank of complex cells and simple cells. I should mention that these complex cells and simple cells, the basis functions here don't have to be Gaussian. They're actually learned from the network. These basis functions are coupled in pairs, and they have to be orthogonal, but the network could learn to fit anything here. These get sent essentially into a pooling stage with a hidden layer, and then that predicts the output. So basically, we're going to model every neuron that we record as a linear sum of simple and complex cells that are rectified together and summed. And that kind of model works pretty well in all of early visual cortex and in MT, as we've seen in previous work from our lab. This is a high parameter model, but we have a lot of data, and we are very careful not to overfit this model. And this is the results of the model. You can see on the horizontal axis here is correlation coefficient, and the blue histogram here is our predictions in a cross-validated data set using a standard deep neural network, and the green is the cross-validated predictions using this hierarchical convolutional energy model. And you can see that this model works really, really well. You might say, well, wait, it's only predicting at 0.8. Remember, we're predicting here at the frame rate of the video, which is we're predicting 16 millisecond bins. In 16 milliseconds, a neuron can only have between 0 and about 4 spikes. And those data are not Gaussian. They're Poisson. So actually, if your correlation coefficient is your metric, 0.8 is basically the noise ceiling. We're predicting, essentially, with this model, all of the predictable variance in these neurons in V1 and V2. So my view is, like, on V1, finally, after 30 years of my life, this V1 model is basically just done. If all you want is a model that's biologically plausible, because all the elements in here are biologically realizable, and you want a model that predicts perfectly, this is the model. And on the bottom here, I'm just showing that this model predicts better than all of the other models that have been proposed by our evil competitors. No, that's our friends, but just from different labs. All right, so now we're going to, I went the wrong way. OK, so this shows you some receptive fields. Here are V1 receptive fields. They look like V1 receptive fields. Right? These are V2 receptive fields. They look a little wonky. The size here doesn't really mean anything because the sizes of the frames here have been normalized relative to the receptive field size. So don't worry about the size here. See, all these V1 cells look like spatiotemporal Gabors, which is great because that's what V1 neurons are. And there are non-classical receptive fields here, but they're subtle. You can maybe see them a little bit on the edges here, like this one's a little curvy, you notice, on the end. This one's also slightly different than the non-classical receptive field, but these are relatively subtler effects in the receptive field. These V2 neurons look like spatiotemporal Gabors, but they can be a little bit more complicated, especially, you notice this one seems to be pretty strange. But this talks about V4. So what do we do in V4? Well, Mike Oliver, the person who did this work, had this brilliant idea. Maybe, basically, you've got a V1, and then to build a V2, you stick another V1 on top of V1, and it takes the output of V1, and it does V1-y things. And then, to make a V4, you take another V1, and you stick it on top of V2, and you do V1-y things to the output of V2. So basically, this whole model is just a hierarchical stack of Gabor wavelet energy filters stacked on top of one another in this hierarchical network, allowing complicated pooling to sort of leverage the statistical relationships over increasingly larger spatiotemporal distances to create more complicated receptive fields. So in this case, we have to take, because V4 receptive fields are fairly large, and V1 has a log polar map, we take a log polar transform on the front end. Don't worry about that. Then we have essentially our V1 model, we have our V2 model, and we have our V4 model. Now, some of you know, Mike likes to call this a scattering module, V2 a scattering transform. Some of you may know about the scattering transform. It's basically just a way to take a bunch of disparate data sets from a lot of different basis functions that are scattered over a very wide range and pool together the information that's relevant in order to create an invariant representation that sort of parcels out all of the variable stuff. And of course, one of the big goals of the visual system is to make an invariant representation. So we have essentially a V1 that feeds into another V1. Then there's just a mixing module that feeds into a pooling module. So all of this stuff is, again, just learned from the data. And now we can look at the predictions in V4. You notice these predictions in V4 are not as good as the ones in V1. But I should mention, if you take any standard classical model of V1, like the Dogman model or any Gabor filtering spatiotemporal model that doesn't have all of the complicated non-classical receptive field stuff in it, and you run it over natural images at a 16 millisecond frame rate, you will get a distribution that looks almost exactly like this. So my take-home message is our current V4 model is as good as the classic V1 model that we all walk around with all the time thinking V1 solved, even though the correlation between the classic model and the responses at a 16 millisecond time frame is only about 0.5. So since we all think that's the glass is half full, I am now ready to say the V4 glass is half full. And the problem is, remember, whenever you're fitting a model, the amount of data you need to improve your model, remember, goes up as square root of n. So if our n is 1,000,000 frames, we're going to need a huge number of frames in order to build a better model of V4 than this. And I doubt that anybody's going to collect that data anytime soon. OK, so what do these V4 receptive fields look like? They look hideous, hideously complicated. They all look, if you notice, kind of like weird AM spatiotemporal Gabors modulating other AM spatiotemporal Gabors, right? So here you can see this is one AM pattern at a low frequency, and this is another one at a higher frequency. All of these look like some weird combinations of AM, except this one. This one is very strange. I should mention previous results from my lab and other people's labs showed that V4 cells, if you wanted to cluster them into two clusters, you would end up with a texture cluster and a contour cluster. And these are probably more contour neurons, and this is more of a texture neuron. OK, these are all hideously complicated. One way you can try to interpret these things, yes, question?
They look a lot like a migraine aura.
Migraine auras, yes. Migraine auras actually usually are V1 based, and we know that based on the retinotopic organization of the migraine auras. But yeah, I can see that, or they could look like a bad acid trip or something like that. Yeah. Yes. All right. So all right. So this now in this case, in this particular video, Mike is fading between the best 1 second piece of space-time for a neuron, in other words, the piece of space-time that evokes the highest neural response, and the receptive field. And this is to give you a sense of what the relationship is between the receptive fields and the stuff in the images that triggers that neuron. And what you will quickly see, or slowly see, when you watch this, is you cannot look at the receptive field of the neurons and have any idea whatsoever of what is going to trigger them in the world, other than if there's a receptive field here, there has to be stuff here. But you won't know what that stuff has to be. There's no obvious intuitive relationship between these. And that is frustrating, but I think it's just inevitable. These neurons live at this intermediate level where we don't have any language to describe the pieces of shape that they represent. We as humans can talk about really simple things in shape. We can talk about color, we can talk about an edge, we can talk about a curve, and we can talk about complicated things like animals and people and phones. But we don't have a language to talk about like the section of space that encompasses half my face and the stuff behind me and my shoulder here. That just doesn't exist in our language. But these neurons have to code for that kind of stuff. And in fact, remember in V4, the attention effect, the top-down effect, is only about 20% of the neuron's response. So 80% of the neuron's response doesn't have to do with image segmentation. It just has to do with the stimulus statistics. So these neurons are really largely driven by the stimulus, and they're coding the entire visual field in a really complicated, hideous way. So we have 800 neurons, so one thing you would like to do is look through all these neurons and figure out what the clusters are. After all, neurophysiology always boils down to butterfly collecting. I have 800 neurons. I want to know, do the neurons form groups? Do they form a continuum? What is the manifold that they all live on, right? That's a butterfly cataloging problem. So we have 800 butterflies here, but each one of these lives in a model space of thousands of parameters, and there's no clustering algorithm that can cluster 800 points in like a 10,000 dimensional space. So we use brain-based clustering, which is we stared at the data really hard for quite a while. I'm not proud of this. It's just what we did. And after a long time, Mike Oliver, who spent years looking at these data, decided there were contour cells, curvature cells, temporal contrast cells, and these AM modulation cells. And that's our provisional cataloging of the kinds of phenomena, but I hope I made it clear you should not believe this as any kind of valid clustering. It's just gives you a sense for what we see. Garrett, you're looking particularly suspicious. Do you just have resting skeptic face, or do you have a question?
I was thinking AM meant like he saw these in the morning or something. But you meant amplitude modulation.
Amplitude modulation, yes. All right. OK. So one question that often comes up when I give this talk is, this seems like a huge amount of work. Why don't you just use a deep neural network like any computer scientist would use? That's so easy. We get the cafe network. It gives us features. You take your stimuli, you project in the cafe network. It gives you activations. You take a linear sum of those activations. You predict your V4 neuron. It'll work great. It'll be way easier. Well, OK, so let's try that. On the horizontal axis is VGG. And on the vertical axis is this convolutional energy model. And you can see if the signal to noise is low, which is down here in the lower left corner, yeah, you can use a deep neural network, and it'll work just as well as this complicated, biologically informed deep neural network. But once you get up to decent signal to noise, in other words, my data is not random, I have actual real signal here, then our network always works better than the cafe network. So, and this network has far fewer parameters than the deep neural network. Our network has tens of thousands of parameters. Deep neural network has millions. OK, and I should just want to give a shout out to Mike Lewicki and Jan Karklin. In the early 2000s, they came up with this interesting model for V4. This model is actually Bruno Olshausen's sparse coding model. Was that me? Bruno Olshausen's sparse coding model with another sparse. It's my pacemaker. No. All right. So it's a. I'll just try to ignore it. It's entertaining. There's a Bruno Olshausen's sparse coding model with another sparse coding model on top of it. Gives you complicated receptor fields look like this. So that's, again, the idea of you essentially have a V1-like thing that does sparse coding of natural scenes. And you take that output and you send it into another bank of filters that does sparse coding of those inputs. And you get these higher order, if you notice, amplitude modulated kinds of patterns. And that's essentially what we're seeing with the same kind of actual not physically instantiated, but a model of neurophysiological data. So I'm pretty happy with that. And I think this is a good inspiration. OK, so to make a long story short, because I'm in the interest of time, if the classical V1 model is correct, then we have a V4 model that's correct. And I'm not going to do this anymore because I've already got 1 million frames of video per cell, and I'm not going to collect 10 million frames of video per cell. That would be crazy. So I'm going to declare victory and move on. So let's move on to something that has its own problems that are different problems from neurophysiology. The problem with neurophysiology is always, how can I get more neurons, right? The problem with humans is, how can I get any data at all? So in my lab, the human method that we use is fMRI. As Garrett mentioned, I'm not a big fan of fMRI, and that's because I like to think that I'm a neurophysiologist, and fMRI does not measure neurons. It doesn't even measure anything next door to neurons. It measures the plumbing, right? MRI is a metabolic signal that measures oxygen use over time. No matter how many times you hear an MRI talk where people say they're recording from neurons, they are not recording from neurons. And in fact, the link between MRI signals and neurons is obscure, complicated, and something you really don't want to think about if you don't have to. So here we have a brain. The brain's inconveniently folded up inside the head. So as you saw, we computationally inflate it and we flatten it out. So in the middle here is the visual system. This is prefrontal cortex left and right. This is the motor and somatosensory strip. And this is the auditory system down here. And now somebody's watching this movie while fixating on a small green dot that's not shown here. And we're simply painting the metabolic brain activity on the surface of cortex while they watch this movie. There's a linear fading here, but it's only linear interpolation between the samples taken every 2 seconds, so there's no data imposed on here. These are the actual data we have. And you can see that as people watch this movie, we get fairly complicated patterns of brain activity, right? Sometimes some things are activated, other times other things are activated, and the patterns of brain activity are constantly changing over time and changing and evolving. And of course that makes sense because after all, anything that you can see in this video must be represented in brain activity, right? And anything that you are reminded of in this video must be represented in brain activity. So I just saw coffee, and I would really like more coffee right now. So something in my brain that said, hey, maybe I could get a coffee, like if I reach down here into my backpack and get my coffee, then that must also be represented in brain activity, right? So some of this brain activity is going to be deterministic because it's going to be directly from the video, and other brain activity is going to be non-deterministic because there's something in the brain that's driving it, but we don't have access to those variables. They're latent variables. Okay. So you can think about MRI as a giant multiple regression problem. I've got stuff in this video and latent variables I can't measure. I've got 100,000 points on cortex I measured, and I just have to solve this giant multiple regression problem for every point on cortex, figuring out what it liked in the movie or what latent variable it responded to. And that's, again, just a system identification problem, except I now have 100,000 black boxes I have to deal with. So for those of you who don't do MRI, this is the only slide you ever need to think about in MRI. This is one slide summary of MRI. It's a bulk chemistry experiment, except now instead of having a chemical, liquid chemical, we have you. We stick you in the MRI machine, and the MRI machine is a big stationary magnet that aligns some vanishingly small fraction of protons along the main magnetic field. Then we introduce an electromagnet. The electromagnet puts in an orthogonal signal, and it spins the protons off axis according to whatever gradient we put in. Now the protons are spinning off axis. We turn this electromagnet off, and now the protons spin back down to align again with the main magnetic field. And we can measure the proton resetting back down to the main magnetic field in terms of the spin rate changing in the transverse plane or the plane along the B0 direction, the main magnetic field direction. And that gives us two parameters called T1 and T2. And all of MRI is basically just looking at these T1 and T2 parameters, these relaxation parameters. And instead of using bulk chemistry, MRI people are incredibly ingenious at coming up with essentially gradients, frequency and phase modulated gradients that they can impose on this bulk sample that essentially tag the spatial location. And they allow us to pull out the dephasing T1 and T2 components from different locations inside this volume instead of just getting the whole volume at once. And so we can get various kinds of weightings from MRI. We can get a T1, here's a T2, we can get PD, or we can get a functional MRI. And a functional MRI takes advantage of a bug, although in this case it's a feature, which is that deoxyhemoglobin has a low bold signal, blood oxygen level dependent signal, and oxyhemoglobin has a high bold signal. And so as the blood flow and the oxygen levels increase, it changes the bold signal. And neurons are little metabolic engines that make ATP, and when they make ATP, they have to extract oxygen and sugar from the bloodstream. The bloodstream has this Rube Goldberg mechanism that when it detects that oxygen has been extracted, increases blood flow, and that's what we measure on MRI. So now we've got these MRI signals. They're these bold signals that are kind of vaguely related to the net synaptic activity in a local area. And now we want to model the data, right? Our input data is complicated stuff, and our output data is this bold signal. How are we going to model it? Well, it's a regression problem. So this is, turns out all this data is Gaussian by the time we're done with it and start processing it. So we're going to use ridge regression. And so we have some features and some bold signals, and we're basically just going to fit these using ridge regression, and we're going to get essentially voxels by feature weights matrix out of this thing in the end. And the cool thing about this method is we can basically test multiple hypotheses simultaneously. And we do this by a process called linearizing or linearized regression, where basically we take our original stimulus, in this case it's a story, and we project it into different feature spaces that we hypothesize might be related to the bold responses that we measured. So for example, we can take this story, we can extract the spectral signature from the story, we can extract the phonemes, we can extract the syntax, we can extract the semantics. Now each of these feature spaces might be larger or smaller. Like to take the sound spectrogram of a story, that might require 3,000 features. Phonemes, there's like 65 or so phonemes in the English language. Syntax, maybe we might use a HHMM with maybe 30 syntactic states for that. Semantics, maybe we'll model 2,000 semantic concepts. So we're going to break the stimulus simultaneously up into all these feature spaces. Then for each one of 100,000 voxels, we're basically going to fit all of the weight. We're going to find beta weights for all these feature spaces simultaneously. And now we're going to collect a new data set with the different stories, and we're going to project those stories into these feature spaces. Again, we're going to multiply them times the weight we got in the first stage, and then we're going to just compare the predicted responses to the observed responses. Some of these beta weights will be large, and we think that's maybe what that voxel represents. Some of them will be small. The voxels will tell us what feature spaces they care about. So this is a way of taking a complicated naturalistic stimulus and simultaneously testing many, many different hypotheses about stuff in the stimulus that could have been represented by the system. And if you notice, this is very similar to what we did in the last experiment, except I suppressed all the stuff about feature spaces there. So now we can look at multiple feature spaces simultaneously. These are data from people listening to a story in the MRI machine. This is 3 feature spaces. And we can, because these feature spaces are correlated, we can do variance partitioning to look at the part and partial correlations. And we can find, for each individual voxel, how much that voxel's activity can be predicted from either the individual feature spaces or the joint feature spaces between them. And the sad thing about MRI is it's very slow because it's measuring this metabolic brain activity. So you can't pull out data about fast changing things like the sound spectrogram. The signal to noise for finding sound spectrogram representations in the auditory belt in humans, that sound spectrogram data is very poor signal to noise, and so we can't really do a very good job of that. But what we can do a good job of in MRI is things that are slower, where slow means on the order of a second or two. Basically, there's very little data, there's very little information in the MRI data at a time scale faster than about half a hertz. So it turns out that semantics, which is the meaning of the stories, has a lot of signal to noise in MRI because the meaning of a story evolves at a time scale that is commensurate with the thing we're measuring, which is this metabolic signal in the brain. So let's look at semantics for a minute. Now, in this particular experiment, our semantic feature space, which we got by latent semantic analysis, for those of you who care, had about, say, 2,000 semantic basis vectors. So we have a 2,000 dimensional semantic space, which spans all the English language. And now we can analyze the brain data. The brain data is just a set of voxels with beta weights in the semantic space. So that means we've got 100,000 voxels. Each of these voxels is represented by a 2,000 dimensional vector. That's a lot of data, and no human can possibly think about that data. So you got to do dimensionality reduction. If I go to give a talk somewhere, the easiest method of dimensionality reduction to talk about is principal components analysis. So let's take this data and do PCA on it. When we do this, if you do principal components analysis on MRI data, you're going to find that about, usually in a typical MRI experiment, it takes about in a typical MRI experiment, where you take a feature space, like this semantic feature space, you'll need about 100 principal components to account for that data. And about 10 of those will be shared, and they'll account for about 30% of the variance. So this just shows the first 3 principal components, just because then I can conveniently show them on a screen where I color each principal component R, G, and B. So if that did not make sense to you, just think about it this way. There's a principal component space where we can project, in which we can project all the semantic concepts. And nearby concepts in this space are concepts that are represented similarly in the brains of all the subjects. And that turns out to be social things are represented similarly to emotional things, for example, and visual things like bumpy, round, and so on are represented similarly to tactile things like rough and smooth. Right? And then we can also project these principal components onto the surface of the brain. And similar colors on the surface of the brain are locations in the brain that represent similar semantic concepts. So all of the red locations on this brain are representing the same stuff. And to see what that stuff is, we can go up here and see what's red. And you can see that the red things are social and mental things. Okay. So this data set's really complicated, and that's not what this talks about. So I'm not going to go over it in detail. If you are curious, you can go to our website and play with this online brain viewer. This is really cool JavaScript application that allows you to interact with the data. So you can click on an individual voxel, and you can find out in this cloud at the lower right what semantic concepts this experiment predicts that voxel would respond to the most. So you can see that all these little red locations respond to social things like mother, father, brother, sister, and things that happen to social groups like marriage, death, divorce, murder, which comes up a lot. Then you can, and there's a bunch of these red spots, right? Now in a second, you'll see that I'll go over here to this green spot. This green spot is actually a known brain area that is a number-related area in posterior parietal cortex. And in fact, there's a whole network of number-related areas. It turns out that these number patches tend to represent numbers, time, money, and weights and measures and dates. All those things are represented in the same network. And there are multiple, again, 20 or 30 different areas that all represent numbers. So there are these multiple networks in the brain for different semantic categories. You can also play with this, another fun tool we have online that allows you to basically go in and look at a subspace. So if you want to find out how the concept of dog is represented, it'll show you these red areas are locations that represent dog, and blue areas represent locations that don't represent dog. To make a long story short, in this semantic experiment, what we find is that every semantic concept, like dog, is represented across multiple brain areas. And every brain area that represents a semantic concept like dog will represent a constellation of related semantic concepts. We think that these multiple representations are due to the fact that the brain needs to represent different attributes of the semantic concept. So for example, you might have an area down here near olfactory bulb that represents how a dog smells. And you might have this area over here, which is near auditory cortex, might represent how a dog sounds. And an area over here on the medial wall might represent where you see dogs in the environment, like in a doghouse, for example. And somehow or other, when you think about the concept of dog, all of this information is aggregated by the brain and emerges into this magical process we call consciousness. Okay. These semantic network interacts with all of the sensory networks. So the semantic network that I showed you in the last slide is pretty much amodal. It doesn't really care whether the information is coming in by sight or sound or smell. But you can actually, once you collect a large amount of data across individuals, you can look for the correspondence between semantic maps, say, for vision with this amodal network. And to make a long story short, I'm just going to show you this slide on the lower right here. This little line is the border between occipital cortex, which is driven by movies and not stories, and polymodal cortex, which is driven by stories, is in blue. And you can see that on both edges of this line, the semantic concepts align almost perfectly. So there's a whole constellation, many different higher order visual areas that respond to certain categories of semantic things in movies, like dogs and cats and buildings and planes and everything you can imagine. And those are arrayed at the anterior border of occipital cortex. And those feed into another network, which is this amodal network that is activated by stories about dogs and cats and planes, or movies about dogs and cats or planes or whatever. You had a question.
Sure. On the previous slide, just curious about why there would be nothing in the ventral stream for dog.
There is. It's just back here and here.
Oh, I'm sorry. Go ahead.
So one thing to remember about the ventral stream is MRI has its own issues. And one of the problems it has is dropout. And the places you get the worst dropout are anything near the ears, which is a bunch of the temporal lobe. You have really low signal to noise. And stuff in the medial wall tends to be very far from the antennas that are receiving the signal, so you have low signal to noise. And stuff at the bottom of the brain, next to the cerebellum, and stuff next to the ventricles, stuff in orbital frontal cortex. All those areas are areas of dropout in MRI. And if you want to target those, you need very specialized pulse sequences that will actually only half compensate for the dropout. So things near the ears, which is probably somewhere around here, we don't have good signal to noise there. So if you don't see something, MRI is like every other method of measurement. If you don't see something, just don't think about it. You're not allowed to say anything about it. If you see it, fine. If you don't see it, you can't say anything. Okay, so that's true for this too. All right. People oftentimes want to say things about things they can't see. Don't make that mistake. All right. So I should mention that this method of system identification for MRI, you might think, well, you know what is this good? Is it bad? I don't know. You didn't test a hypothesis. What do I think about this? The one thing I want you to remember is we were optimizing for information flow through the system. And this method, the easiest way to see how much information you gain from a method is to build a decoder. Building a decoder is a bad way to do science, but it's a really good way to visualize how much information you recovered. And this method recovers way more information than all the other methods in MRI by log units. So these are data from one movie experiment. We put people in the MRI machine, we showed them movies. Now, this decoder on the left, which is these 4 panels, is a decoder that was built using essentially a motion energy pyramid, kind of like the one I showed you for V4, where we're decoding information only from primary visual cortex. And on the upper left here is the image we showed. On the upper right is our reconstruction. And just to make it clear how good this reconstruction is, we have the edge maps here on the bottom. You can see that the edge maps align pretty well. On the right is a decoder we built using the same data set from another couple of visual areas that are higher order visual areas that represent the semantic information in the movies. And you can see that this decoder also works really well. We're decoding about 300 concepts simultaneously. Each TR, each 2-second period, we can decode about 8.5 bits of semantic information using this one model. So you figure there's 400 or so brain areas in the human brain. We could build an optimal decoder for each one of those brain areas and decode all of that information simultaneously if we wanted to. There's a lot of information in MRI. It just has to be used correctly. So in the last couple of minutes, I just want to mention one of the other cool things about this method is that it allows you to get maps in each individual brain. So this is one semantic map. This is another one. These are 4 semantic maps from 4 people. And you can look at these semantic maps and you can decide, are they similar or different? Now, I won't waste time taking the poll because I've done this many times before. A lot of you are saying, these maps don't look the same. And you're 70% right. In these data, about 30% of the variance is common group variance, and about 70% of the variance represents individual differences. And if you look, everybody has 2 red spots in the temporal junction. 2 red spots, 2 red spots, 2 red spots, 2 red spots. Everybody's got some green stuff above that red spot. Green stuff, green stuff, green stuff, green stuff, right? Everybody's got a big red patch with a blue patch up here in the medial wall. Red patch, blue patch, red patch, blue patch. Right? You can see there's commonalities, but there are also differences, especially in prefrontal cortex. You can see this is a particularly nice brain down here. You can see there's a orangey stripe, a pink stripe, a blue stripe, orangey, and then some spots. And everybody kind of has that orangey, pinky, bluey, greeny, right? So these are, you can see that these are kind of similar, right? Now, the way we deal with this kind of data in MRI, sadly, is we average. So this is an example, just in case you don't do MRI, to give you a sense for what happens when you average. So imagine you have a bunch of presidents, and you decide you want to map them all into some generic president space. So your way to do that is just average the heck out of them. This is actually intelligent averaging because they take basically key points from the presidents and they align them all together, right? And people do that in MRI. They take key points like the soul sign gyri and they align them. People aren't idiots. They do the best they can with this method. But still, you see that this average president, not only does it not look like any real president, but also there's these weird artifacts and it just seems blurry. And the reason it seems blurry is you can't perfectly average presidents together because it's a non-convex optimization problem. You don't really know what the right answer is. So you're going to do some hacks, and anywhere you did a hack, it's going to be suboptimal smoothing. Optimal smoothing is great. I love optimal smoothing. I hate suboptimal smoothing because that means you lost information, right? So this is going to do suboptimal smoothing. In every place it's suboptimally smooths, you're going to lose information, and that's what happens in MRI. And here's the example of that. Here are 8 individual brains. We're just looking at the occipital lobe here. You can see the data from these 8 individual brains. And here's a cross-subject average we got by using the best possible method for cross-subject averaging that you can get in the standard MRI software packages. And you can see that a whole bunch of the information from these individual brains is wiped out when you do that. So that's bad. We don't want to do that. We want to basically have a model where we have individuals and we have a group and they're related to each other, but when we go from the individual data to the group, we don't just blur the heck out of everything and lose all the information, because I told you at the beginning of this, I don't like losing information. So after many years of hitting on this problem, we come up with this new generative model for dealing with this, which I really like. It has the awesome acronym PRAGMATIC, which is probabilistic and generative model of areas tiling cortex. And I should mention up front, that my contribution to this whole project was I came up with the acronym pragmatic. Okay? Everything else in this project was my grad student, my awesome grad student now at Texas, Alex Huth. And he developed this model. He said, well, let's think about this problem as, a cortical sheet is kind of like a pizza, where each of the functional areas is a little pepperoni on the pizza. Now, if you go into Domino's and you tell the Domino's manager tells the new trainee, okay, I want you to make all the pizzas identical and everybody gets 16 pepperonis. Well, that guy's not going to make all the pizzas exactly the same. They're going to be kind of mooshed around, and so the pepperonis are going to slightly shift on every single pizza, right? And that's kind of the problem we have with these functional areas. So he said what we can do is we can basically learn a division of the brain into this functional areas from this continuous data. We can basically fit a ball and spring model to allow for the individual differences, and we can learn those ball and spring parameters. And then we can basically build a generative model that, given a new piece of anatomy, will generate the functional patterns for that data. And this is really cool because nobody had done this before. So here are 3 subjects done this way. Now I should mention, in each one of these rows, this subject was left out of the pragmatic algorithm. So we took the functional data and the anatomy for, say, 7 subjects. We trained it on pragmatic, and then we predicted where the functional data should be for the new subject. So here's the subject's true functional map. Here is the pragmatic parcellation, and these are the likelihood ratios. You can see that in all 3 cases, pragmatic does a really good job. Red indicates locations that are semantically selective, where the pragmatic model is correct. White means it's not semantically selective, so it's irrelevant to the model. And blue, you notice there are some blue areas, and blue areas are areas where the model predictions are wrong. And these are always areas, if you inspect this data closely, where it knows there is a functional area that's supposed to be at that location, but in the real person it's slightly shifted off from where it's supposed to be. So this is a really cool method that we're developing. It's not ready for human use right now. Alex is the only one that can use this software, and he's a mutant. So I'm pushing him really hard in his new lab to make this into a software that everybody can use. You can pull out now a functional network. This is a functional parcellation for semantic information that pulls out like 150 brain areas from one experiment. You don't have to do 150 experiments. You get all the brain areas from one experiment, and you can now use this to build optimal localizers to identify each one of these areas, whichever one you want. OK. Do I have 2 minutes at the end? No? Yes? No? I can quit. I can quit. 2 minutes? I don't know. Leave if you don't want to. This should just be quick. This is the last thing, because I just want to mention, because I know there's some clinical people here. What you want to do is, what you would like to do is use this method in the clinic. For those of you who don't know, MRI is never used in clinical applications except for pre-surgical mapping for epilepsy. Because if somebody has epilepsy and a doctor is going to cut out a part of the brain, they want to make sure it's not a brain that you will notice is missing. So they map eloquent cortex before they do the surgery, motor cortex, to make sure that they're not damaging anything that they can avoid damaging any more than they have to. What we would like is to move this method into the clinic so that we can map individual people that have certain neurological disorders, like somebody may be preclinical for Alzheimer's or some other neurodegenerative disease, and, or schizophrenia, or a wide variety of other mental disorders. If we can use these high-dimensional mapping methods, then we'll get way more information from each individual person, and we'll be able to maybe actually make MRI clinically useful. So the problem is, our experiments in our lab take, individual subjects are in the MRI machine over a period of weeks for, somewhere between 3 and 6 hours, and that's not clinically realistic. You have to be able, in the clinic, to get your data in 20 minutes. Because if it's not 20 minutes long, it doesn't fit in the MD's workflow, and they won't do it. So we have to try to get the same semantic maps we got out from a 6-hour data set from a 20-minute data set. And that means you want to optimize the experimental design, you want to optimize the model. So we started optimizing the model by doing the following. We built an autoencoder. This autoencoder is called a multi-view autoencoder. It's a certain kind of autoencoder. And in this particular case, our views are different brains and different feature spaces. And we basically take all the data we have from a bunch of subjects with different brains and different feature spaces, and we shove it through an autoencoder. If you remember, an autoencoder is the best unsupervised deep neural network learning method we have. An autoencoder goes from a big feature space through a little tiny feature space back out to the big feature space. And by doing this, by having the output layer and the input layer, the cost function is basically that this output has to match this input. So essentially, the thing learns to match the output as closely to the input as possible, given the constraint of this hidden layer here, right? So it's a really good method for dimensionality reduction. So we put all these brains into this autoencoder. We train the autoencoder. And the cool thing about a multi-view autoencoder is you can now take a new brain. The system has no information about this new brain, and you can predict on the output end what this new brain should look like. And just to give you the last slide of this talk, here is data from one subject from a narrative comprehension experiment. Here is data predicted for this subject from this multi-view autoencoder. This data set at the top was collected from 3 hours of people listening to stories. The data set at the bottom was collected from 20 minutes of this same subject watching movies. And none of the story data was used to build this model. But you can see that this model does a really good job of predicting this data. So we're very optimistic that this might work in the future. And I think that's about it. Thanks very much for your time.
I don't see anybody pushing on the door for us to get out. So we have time for maybe a couple of questions.
Nobody has any questions? Oh, okay.
Question? I have a question.
Yes.
So
One guy has a question.
The fMRI mapping you showed at the end was, I think, almost completely cortical in nature. Any thoughts on sort of pushing that to other brain structures?
There are many people who use fMRI for subcortical mapping, and I always feel sorry for those people. So look, MRI is like it's just radiochemistry, right? There's an antenna and there's a sample. And basically, this sample is going to emit some energy. Basically, in this case, I'm going to put energy into the sample, and then I'm going to turn it off, and the spins are going to precess back down, right? And it's going to emit energy, and I'm going to measure that energy on the antenna. So like any simple system, this follows the inverse square law. If your sample is really near the antenna, the world is great. You get high signal to noise. The farther your sample is from the antenna, the lower your signal to noise. So the antennas are on the outside of the head. The bowels of the brain, the giblets of the brain, are like really far from the antennas. So the signal to noise for cortex is awesome. The signal to noise for anything far from cortex is bad. And then on top of that, you end up with a resolution problem, which is, MRI, functional MRI nowadays, the typical resolution you're going to be able to get is 2 to 3 millimeters. And if you take the whole hippocampus in a human, that's going to be maybe 5 voxels at best. So you're hitting up against multiple problems. Now, Berkeley is actually building the next generation of human MRI for cortical mapping. So it's going to be a 7 Tesla stationary magnet, but all the guts of the magnet are being completely re-engineered from scratch. And that should take us, for cortical imaging, from 2 to 3 millimeters down to 400 microns. And that's a good number because a cortical column is about 500 microns across. So that if this very expensive physics project works, we'll be able to actually record column-level, non-invasive data from humans, but only from cortex, because the antennas are going to be very small and the gradient's very strong, and so you'll have really good resolution right next to the antennas. But once you get down to the giblets, it's not going to be any better resolution than any other MRI machine.
Other questions? Comments? I have another question then. You mentioned, was there a question? You talked about representation of the different voxels. I'm just curious if that representation changes over time, for example, if it's context dependent. What happens?
Let me see if I have a slide on that. So if I don't have a slide, I can describe this. Oh, I do. But I don't think I have the right slide. Yep, I'm not in the stack. OK, so attention, the traditional way to think about attention is that it changes the gain of neurons. Okay? But the thing to remember is if you have a deep network, say you have just the cafe network, and you change the gain of a small number of units on the peripheral edge of this network, that will actually have the effect of changing the tuning at this higher level. It'll change the features that are represented at this higher level. In fact, that's how you train a deep neural network, right? Deep neural network starts with random weights, and then you change the weights by very small amounts, and that changes the actual representation at all subsequent levels. So it's almost impossible to build a deep neural network that doesn't have the consequence of small gain changes at one level changing tuning at a higher level.
So the language we usually use is about some area of the brain or neuron or whatever having some representation, but
it's not fixed.
It's not the right kind of language to use for this because the representations will change.
Again, I think it's like, this linear approximation we all make because humans aren't that smart and the brain is. And the thing to remember is it's a fiction, right? So we first showed tuning shifts in V4 neurophysiology back in 2006, where we had animals attending to streams of stimuli, and you would basically, these stimuli were coming in very, very fast. You tell the animal, pay attention to the dog, pay attention to the cat, and you would see that the tuning curve for an individual V4 neuron would shift toward the dog or shift toward the cat, depending on what he was attending to. But the amount of shift was small. It was only about 15 or 20%, because that's all the attention effect you get in V4. So we did an experiment in 2013, which I'm still surprised works. This is an experiment I tried to get people to do in my lab for 2 years, and no one would do it because no one thought it would work. And then I had an MRI physicist come to my lab. He didn't know anything about neuroscience, so I could convince him to do experiments that no one else would do. So I was very happy with this because it worked. We basically had a person in the MRI machine. We had them watching movies, and in condition A, we said, look for vehicles. And every time you see a vehicle, you hit the button. And in condition B, look for people. Every time you see people, hit a button. Okay? So now we have two different attentional states, and we can model the entire brain, the visual semantic selectivity for the entire brain under condition A, searching for vehicles, and in condition B, searching for people. The reason we picked those two semantic categories is they're kind of the most dissimilar categories you can pick if you're thinking about brain representations. And what you see in that case is that voxels all over the brain will shift toward the attended category. The tuning of the voxel shifts toward the attended category. But there's two interesting aspects of this. First of all, the amount that the voxel shifts toward the attended category is inversely proportional to how far it is from the sensory areas. So V1 voxels shift almost not at all. Voxels in prefrontal cortex will shift completely. And that's also consistent with the monkey data we have from Earl Miller's lab, showing that prefrontal cortex neurons are basically completely determined by the task. They'll either respond or not respond depending on the task. So the other interesting thing we found from that experiment was that most of the brain shifted toward the attended category, but there was one network in the brain that shifted away from the attended category, and that's the default network. So if you search for dogs, for those of you who don't know the default network, right? If you put something in an MRI machine and you tell them don't do anything, this network of areas in the brain becomes active, and it's called the default network because when people who first discovered this weren't psychologists, and they thought when you told somebody to not do anything, that they were actually not doing anything. It turns out that the default network is actually the rumination network. And if you put somebody in the MRI machine and tell them not to do anything, they start saying to themselves, when the hell do I get out of this MRI machine? This is really uncomfortable. I really want to have a beer. Where's my $50? Why? They talk to themselves. It turns out if you ask somebody to talk to themselves in the MRI machine, you light up the default network. Okay? So it's kind of the inner directed network. It's the network that's directed to the little person inside you instead of the outside world. So the default network in these attention tasks actually shifts away from the attended category. So I have a just so story about this. I have no idea if this is true. My theory is, imagine you go home tonight and your cat Fluffy is missing. And you all of a sudden start looking, where's Fluffy? Where's Fluffy? Where's Fluffy? Well, you don't want it. You're thinking about Fluffy. You're remembering what Fluffy looks like. You're generating all these images of Fluffy and all these hypotheses about where Fluffy could have hidden based on her past behavior. But you don't want to respond to those internal representations of Fluffy, right? Because then you would say, oh, there's Fluffy, she's in my head, everything's great, right? That would not work well. So you essentially detune the network that is generating this internal speech about the thing that you're concentrating on if you're concentrating on the thing in the outside world. And you tune all of the resources you can toward the thing in the outside world to optimize your allocation of hardware. So it's essentially a matched filter. The brain tries to build as good a matched filter as it can, but it can only build that matched filter based on the connections of the neurons. In primary sensory cortex, neurons only have very local connections, and they can't shift very far toward the attended category. But in prefrontal cortex, because of all the indirect connections through the network, those neurons have access essentially to the entire state space of the entire brain, and they can do whatever you want. And that was way longer answer than you wanted, but I think attention shifts are really important, and nobody studies them. It's really, really important. So anyway.
Well, I'm just happy we didn't get kicked out of here because that was a great story to end this. So, let's thank Jack again.